Good day, students. You're welcome to our class on genetics. And in this class, we started with the nucleic acid. And we said that the nucleic acids are how many? If you can remember, there are two. We have the DNA and we have the RNA. And we said that what forms the building block of the nucleic acid is what? Is the nucleotide. Let's say together is the nucleotide. And the last class, I drew for you the structure of the nucleotide. And we said the nucleotide has three parts. We have the inorganic phosphate, we have the pentose sugar, and we have the nitrogenous base. In the last class, we also talked about the 5 types of nitrogenous bases, and we grouped them into 2. We have the purine bases. Let's remember together. We have the purine bases, and we have the pyrimidine bases. So can you remember examples of purine? Purine are 2, as I told you. So the first one is we have the guanine, okay, and we have the adenine. We have the guanine and we have the adenine. So the purine bases now, the pyrimidine bases are going to be 3, okay. Then, so we have the cytosine. We have the cytosine. So the reason I'm drawing an arrow between the guanine and cytosine is that the guanine always pair with cytosine. Now we're advancing our knowledge from where we stopped in the last class. Last class, I only highlighted the examples for you, but now I am pairing them. Okay, so guanine always pair with cytosine. The next one is thymine. You can see here yourself that adenine always pair with thymine. Adenine always pair with what? Thymine. Then you have uracil. Okay. Now let's go to the structure of a particular nucleotide. Sorry, the structure of or repeated structures of the nucleotide. So, okay, let's start with the DNA now. The DNA. And don't forget that the full meaning of DNA is deoxyribonucleic acid. For the last time, deoxyribonucleic acid. So what is DNA? The DNA is a double strand of nucleotides. Or you can say the DNA is a double helix structure, or we say the DNA is a double strands of nucleotides or repeated nucleotides. It's a double strands of repeated nucleotide, and that repeated nucleotide is called polynucleotide. Poly means many. So when you have a lot of nucleotides, are you there in 2 sites? Then we have what is called the DNA structure. Okay, so let's have the structure of the DNA now. So the first thing is for us to draw the pentose sugar. Please, you need to learn a lot here, and it's going to be very simple. Let's draw our sugar. So let's draw our phosphate here. Perfect. Okay. So the next thing is for us to draw our base here. So this is a nucleotide. This is a what? A nucleotide. Now we need to repeat the nucleotide. Then we now double the nucleotide, and that forms our DNA. Okay. So how do we repeat this now? We have another P joining here. Perfect. We have another base here. Okay. Let's go again. We have another P. Then we have another nucleotide joining. Okay, I'm satisfied like this, so that we don't have too complex structure. Perfect. So this is the pentose sugar. Don't forget, this is your sugar, your pentose. This is your pentose, and this is your pentose. So I need to assign base. I need to assign base to my nitrogenous bases. And I'm going to I assign bases using the letter. So I told you in the last class that the guanine is letter G, the adenine is letter A, cytosine is letter C, thymine is letter T, and uracil is letter U. So I'm going to be starting with guanine. So I'm going to having G here. Okay, let me go with adenine again, and let me have cytosine. I can pick any one of these. Any one of these. The only one I cannot pick is uracil. Uracil is not found in the DNA. So let's highlight. Let's come over here, please. Let's highlight the nitrogenous base for a DNA. Very important. All the 4 bases are found in the DNA except uracil. So we say all bases except uracil. All bases are found in the DNA strand except uracil. That is good. So I can pick any one of these 4 as a starting. So that means that the opposite will now be, if there is G here, then G will pair with the C. If there is A here, A will pair with T. And if there is C here, C will pair with G. So you start with any one of these 4. Okay? So the next thing is for us to draw a bond. Is to draw a bond between, that is going to bond between the nitrogenous base. So I have the nitrogenous base here. So I'm duplicating this in the opposite side. So we have nitrogenous base. So what I'm going to do now is that this is going to be upside down. Okay? At advanced level, when you get to university, you are going to appreciate this diagram. Okay? So this is going to be opposite. So I'm starting here now, and I'm going down. I'm starting here, I'm going. This is the pentose sugar, but it is upside down. Okay. Now, where is my pentose? Where is my phosphate group? So my phosphate group is going to be here. You can see this is different from this. It's the opposite of this. Okay. So the next thing is for us to draw the phosphate group. That's the next thing. We have the P here. We have another P here. So we have this. Okay. Then we have this. Then we have our base here. Sorry. Entering into. Okay. So let's have another P here. Let's have our pentose sugar. Okay. Then another base will enter. So just study. You don't need to stress yourself and memorize. You just study and see what is happening. Okay. So we're having the pentose as usual here. We're having the pentose. We're having the pentose. And we're having the pentose. Now let us pair our bases. Let's pair our bases. We have the guanine here. So the guanine will always pair with the cytosine. And cytosine is a pyrimidine. Cytosine is a pyrimidine. Don't forget that. So we have C is going to be here. So adenine, what does it pair with? The adenine pairs with thymine. So we are going to have T here. And the C, that's cytosine here. You go back to the purine basis. Cytosine will pair with the guanine. So you have the G, and it goes like that in repeated units. Now let us look at, this is oxygen is going to be here, oxygen is going to be here, and oxygen is going to be here. Oxygen is going to be here, oxygen is going to be here, and oxygen is going to be here. So let's look at the carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5 will be here. Okay, where is carbon 1 for this? Carbon 1 will be here, carbon 2 will be here, carbon 3 will be here, carbon 4 will be here, and carbon 5 will be here. Carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. Okay? The carbon 5 is between the line that connects the sugar and the phosphate group. Take note of that. Now, when we end, what do we use to end this? We end this with, what is carbon 1, 2, then 3, 4, and 5. You can see that we are starting with 5, okay? And we are ending with 3. So this is 5 prime and this is 3 prime. What are we starting with here? We are starting with 3 here. This is 3 prime. And we are ending with, let's go 1, 2, 3, 4. Then the P is going to be here now. Then 5. This is going to be 5 prime. Okay? This is not the focus of the exam. I'm only showing you the placement of the carbon. You refer to your organic chemistry where the carbons are being placed. But the focus of this class is that now we are having one strand of nucleotide, and we are having another strand of nucleotide. Okay? They are connected together by their bases in opposite sides. Then what connects each of the nucleotide together is the hydrogen, or the strand together is called the hydrogen bond. You can see the dot 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 is a weak bond. Okay, it is the hydrogen bond. Okay, so this is the DNA strand. Okay, now let us go to the Now, the DNA strand is not as this structure as you have seen. Okay, it is always winding. It's always winding. So let us see how, and forms a double helix structure. This is a structure, but it's not straight as you have it like this. So this is how it forms. This is what will form the DNA. So this is the line. This is the one here. You can see I can even write my prime by saying 3 prime is starting here, 5 prime is starting. Okay, then the nitrogenous bases will be in the middle. Okay, so you continue like this, so which forms a double helix structure. Okay, which forms a double helix structure. If 5 should start here, then 3 prime is going to end here. 3 prime starts here, then 5 prime is going to end here. So this is the double helix structure of DNA. That is a double helix structure of the DNA. So how do we form our RNA? It is very easy to form the RNA. RNA is just one strand. So you delete a strand of DNA to form an RNA. So let's have it as RNA here. So we are starting with RNA now. So what is RNA? RNA is a repeated unit of nucleotide also, but it is one strand. Okay? It's a one strand and it is repeated unit. Okay? So you say it is a strand of polynucleotide. Don't forget that we define DNA as double strands of polynucleotide. So I'm defining RNA as what? A strand of polynucleotide. That was why I deleted the other strands. So we're having one strand. And the bases of RNA are just 4 also. Okay? But we don't have thymine. Uracil will replace thymine. So we say the bases are all bases, all the bases except thymine. All the bases except thymine. We have them inside the RNA. So, do I have thymine here? No. I have guanine, I have adenine, I have cytosine. So let me just draw one again and end the class with that. So we have the P here, another one here. Then we have the sugar here. Okay. Then we have the bases here. So we have guanine, we have adenine, we have cytosine, then we are going to have uracil. So you're not going to see me writing thymine. Okay? This is what is called, this is the structure of the RNA. So how do we draw the strands of RNA? Just very simple. Just very simple. So you have this, you have this, you have this. So this I'm drawing, they are the nitrogenous bases, but they are not connecting to another strand. So this is a strand of RNA. In the next class, we're going to be talking about the difference between the RNA and the DNA. Thank you so much. Goodbye.